Hello, innovators. I hope you guys had a great break before our next speaker, Dr. Philip Heller.
Dr. Philip Heller is a computer science professor at San Jose State University. He currently uses machine learning to analyze how climate change in Antarctica is affecting the food web. He works with the Coral Reef Division of the National Oceanic and Atmospheric Administration and has also been working with NASA to analyze astronaut health risks due to radiation and microgravity. Please welcome Dr. Philip Heller.
Thank you for the introduction and congratulations on a great conference so far. I'll do my best to live up to your standard. I noticed that I don't have any way of knowing how much time has passed by. So if one of the hosts could please let me know when I have about 3 minutes to go, that would be great. I see that there is an error in my first slide. That's not me, that's a cuttlefish. That's me. Okay, now that we've got that straight, I'd like to follow the example of our first speaker and tell you something about my journey of how I got here. Not so much because I'm such an interesting person, I'm not, but I want you to understand why I do what I do and why I enjoy it so very much. So like our first speaker, I got a double E degree, electrical engineering. All that proves is that you can't spell geek without a double E. I moved to Silicon Valley from Berkeley and I wrote a lot of software and I wrote a lot of books about how to write a lot of software. And a lot of my software actually got shipped. It got shipped as part of products that people actually paid money for and people bought my books. So in that sense, I was a success. But there came a point where I looked at my reflection and wondered if I had actually made the world better. This is different from success because success is a value that society invents. And our society says that if you ship a lot of software and make nice money for it, you are successful. But personally, my personal definition of success was shifting. And I realized that I wanted to make the world better. And I had no way of knowing if the software products I had made were making the world better, or if the people who read my books and became better programmers were using their superpowers for good or for evil. And around that time in my life, I learned about a new science called bioinformatics, which roughly speaking is computing about DNA from an environment to figure out what's going on in that environment. I got super hooked by the idea. And at age 52, I applied for a Ph.D. in bioinformatics. The first place I looked for a PhD program was a very prestigious local university that I prefer not to name. And I went and spoke to a member of the program and I said, what do you think? I'm thinking of getting a PhD in this stuff. And he said, well, no, don't bother. You can't possibly succeed. You're too old. So I muttered something underneath my breath that I'm not going to repeat in polite company. And I went to Santa Cruz, which arguably is not quite as prestigious as this other place, but arguably it is where bioinformatics was invented and developed. And I spoke to one of the founders of bioinformatics, a man named Dr. David Hausler. And I said, am I too old for your program? And he said, no, we'd love to have you apply. This was a big life lesson because for me, I prefer to be in a place where kindness prevails. So I went there, and here's something that I learned. Mostly bioinformatics at UC Santa Cruz is about developing very advanced custom cancer therapies. This is great. This saves lives. But the way it's done wasn't all that different from people working in cubicles 14 hours a day, writing software, staring at screens. And I had already spent really over 30 years of my life doing that, and I was looking for something different. So I started looking around at other domains that do bioinformatics, and I started hanging out with the oceanographers. And I realized that this is where my passion lay. because when you're an oceanographer, you actually get to go to the ocean and go be in it. You don't get paid nearly as much as you do for writing software all day in a cubicle. In fact, I figure my salary right now is about a third what I could be making. So in order to decide if I was going to commit to this lifestyle, I did something I'd never done in my life. I took a piece of paper, I drew a line right down the middle, and I wrote the pros and cons of becoming an oceanographer versus the pros and cons of doing more standard computer programming about cancer bioinformatics. And on the left side of my line, I had things like making money, curing cancer, hanging out with interesting people. It was a very long list. And on the right side of my list, I only had one thing. On the right side of my list, it said, dude, it's the ocean. And that convinced me. That was all I needed to know. I made the choice and I've never looked back. If I think about the money I could be making, doing something that I don't completely love, I subtract what I am making, that difference is more or less, I think of it as the money I'm paying for the amount of life satisfaction I'm getting. And my friends, it's a bargain. I'm one of the luckiest people in the world. I love my job. So let's tell you about it. Now, before I do, I want to give you some advice. I want to tell you one of the most important things I learned in PhD school, how to have a permanently fascinating career. Are you ready? Are you taking notes? Be the best, or better yet, the only programmer in the room. I've been the only programmer in a room full of world-class oceanographers. I've been allowed to take on world-class problems. Not because I'm that great a programmer, I'm good enough. I'm not a world-class programmer, but because I'm the only one who is willing to learn about the domain of the problem and be fascinated by it. And to be honest, to be paid a whole lot less than I could be making at, say, Google or Facebook. Most of my career, I was one of many good programmers in a room, and therefore I didn't stand out. But if you're the only programmer in the room, then you're going to stand out, and they are going to give you some really interesting things to work at. Now, another thing I learned, really since in the past 5 years I became a professor here, is how to save the world. Would you like to know how to save the world? You would? Okay. Well, here's my plan for saving the world. It has evolved. My plan began by thinking I could save the world by pretending to be Han Solo. One Jedi, he doesn't know he's a Jedi, but he is. One Jedi, my friends, cannot save a planet. And so I had to change my self-image. I had to, with some humility, realize that the way to do it was to stop pretending I'm Han Solo and start pretending I'm Yoda. One Jedi can't save the planet, but 1,000 Jedi can. And Yoda trains the Jedi. And my friends, some of you are those Jedi. So let me tell you what the Jedi approach to the environmental problems of the planet entails. It entails machine learning. And I just want to make sure that everybody in the audience has just a slight intro to what machine learning is. Here are a couple of machines, and we think of them as learning stuff. The technical definition of machine learning is a bit more precise than that. A machine learning program is quite often a classifier. That's the kind that I work with. And machine learning means that the classifier infers its parameters based on training data. So that means that instead of me, the programmer, having to look at the data and figure out the algorithm for a classifier, for example, what is or what is not an invasive species, what is not a healthy coral reef. Instead of my having to figure out the algorithm, the hard work, from my point of view, is finding really good training examples to, so to speak, show to the algorithm. And the algorithm figures out how to configure itself, how to parameterize itself in order to make the best possible classifier. A nice analogy that I use for people who have no computer experience is based on an article that came out in Discover magazine in 2011 about dogs. You know, dogs have got really, really powerful brains behind those noses. Their olfactory, their smell processing apparatus is very sophisticated. And we've all seen movies where the hounds are going out looking for someone. So you take a scrap of that person's clothing, let the hounds sniff it, and then they go look for that clothing. Here we have a dog sniffing a sample of the breath of somebody with lung cancer. So when there's cancer in the lungs, certain molecules are exhaled with every breath, and a dog's nose is sensitive enough to detect it. And the person handling the dog doesn't know anything about what those chemicals are. That's the beauty of it. We don't need to know exactly what the chemicals are. We just need to show the dog a lot of samples. We call these training samples. And eventually the dog becomes really good at detecting that odor somewhere else. So machine learning, including deep learning, which you've probably heard a lot about. Deep learning often takes the form of convolutional neural networks, which I'm sure you've heard a lot about. And there isn't time to go into what those are. I do want to show you a picture of not a neural network, because I'm going to show you only one. It can't really be a network if there's only one of them. But I want to show you what I believe is the first artificial neuron. Are you ready? Cost 2.6 million in 1958. That's about 22 million today. And here's its inventor, Dr. Rosenblatt. And right here at the end of my red arrow, that's a wire cutter. So he's debugging with a wire cutter. So I'm happy to say that in the years since 1958, things have come a long way. And you can take classes to find out exactly what a neural network is. I'm not going to cover that. I want to tell you about what I'm using neural networks for. So I'm going to tell you about 3 projects that relate to the ocean. The first is about corals, the second is about sponges, and the third is about plastic. It's obvious why we ought to care about plastic in the ocean. Maybe it's not quite so obvious why we ought to care about corals or sponges. I'm a scuba diver. I love looking at them because I find them stunningly beautiful. Catering to my hobby is not enough reason why the world should protect coral and sponges. The reason we should protect them is that we like breathing. And no matter where you live, you could live in central Kazakhstan, 3,000 miles from the ocean in any direction. Even so, every breath that you take contains oxygen that was provided by ocean organisms. And the atmosphere that we breathe is kept in balance by ocean organisms. So let's talk about corals first. One estimate is they fringe about 1/6 of the world's coastlines. That's a lot. And quite literally, they support millions of species. We oceanographers really have no idea how many species there are in the ocean. In fact, we're kind of getting away from the idea of the species concept because it doesn't quite capture all the variation and diversity in the ocean. Millions of species. Biodiversity is what makes an ecosystem robust. When there's a lot of diversity, then when challenges come, like say global warming, some life can always survive. I don't want to downplay the stunning beauty of corals. They look like this, and they harbor life like this. And you can get up close to them like this. Economic importance. Please don't confuse economic importance with importance. Economic importance is only one kind of importance. Mostly from tourism and fishing. The estimated income, just a little short of $50 billion annually. So imagine buying a $1.2 trillion bond and investing it at prime rate. That would be the economic value of coral reefs. They have a biological value because if we destroy them, we only have a few years to live. The ecological importance of coral reefs is that they concentrate biodiversity. That makes them what we call rainforests of the ocean. They're also hotspots of these two processes that make life possible and make the atmosphere possible: nitrogen fixation and photosynthesis. These are the processes where life takes unliving matter and converts it to living matter. And also, if you imagine the shape of a coral, here, let me go back. Say this one. Do you see how convoluted it is? If you're a tiny baby fish, there are lots of places to hide. Most tiny baby fish in the oceans, at some point in their life, go to a reef for safety. So without corals, without reef builders, it's very difficult for fish to live. And one estimate I saw is that at least 1/10 of the world gets most of its wild protein from fish. Well, these reefs are under threat from pollution and destructive fishing and climate change. Before I go on, I want to put your minds at ease. This is not one of those talks where I'm going to talk about how horrible the world is and then leave it in your hands. What I want you to get out of this talk is a sense of hope. But in order to get you there, we do have to talk about the threats that need to be addressed. Climate change mostly means ocean warming. And if you look at this beautiful coral down on the bottom left, actually this coral isn't purple. What's purple are the symbiotic microbes that live on the coral and make it able to be a hotspot of biodiversity. But these guys don't like heat. And when the ocean warms, we have what we call coral bleaching, which is where the microbes leave. And we have what we see over on the right. Ocean acidification is another problem. This is why greenhouse carbon footprint is so important. When carbon enters the ocean, it becomes carbonic acid. And the ocean gets a little bit fizzy. Every time you drink soda, the reason it's fizzy is that it was manufactured in a tank of water, and a little carbon dioxide charge was exploded in that tank. And the carbon dioxide was driven into the water, turned into carbonic acid. It was very weak acid, but it's strong enough to degrade calcium-based shells of anything, of corals, of shellfish, the substructure of the reef. So what we have on the right here is what we call coral rubble. This stuff is dead. Well, the first step in figuring out what to do about it is to study it. When we can look at a coral reef and say, well, that kind of looks pretty healthy, but how do we know? Well, there's a very cheap device called an ARMS, Autonomous Reef Monitoring System. That makes it sound a lot more complicated than it is. It's just a bunch of gray plastic plates bolted together. These are about 1 foot by 1 foot. And you can see these round holes in the corners at the bottom. What you do is you take it and you bolt it to the ocean floor. You'll leave it for about 18 months. And when you bring it back up, it's no longer a bunch of plastic, it's a habitat. And by studying the biodiversity of the habitat of the plates, you can get a sense of the biodiversity, that is the health of the reef itself. And it turns out that this works really well on a very small scale. Even high schools in coastal places with warmer water than us, for example, I know of one in San Diego, are using these in their marine biology classes. And usually you analyze these by unbolting them and just looking at them and writing down everything you see. There are also genetic ways that I won't get into. And it's very low cost at high scale. You just need to find somebody who's willing to look at each plate, take about 10 minutes and tell you what you've got. Trouble is, you can do that once, you can do that 10 times, but you can't do it 14,000 times. NOAA, the National Oceanographic and Atmospheric Association, think of them as being like NASA of the ocean. Their reef assessment monitoring program, over 12 years, deployed 14,000 of these plates, mostly where you see the stars, and they recovered them. And now what? So here's why I told you about my experience of wanting to be the best or only programmer in the room. I heard about this, and I knew about deep learning. I also knew that part of the protocol for recovering these plates is to spread each plate out in the sunshine and take a high quality photograph of it. And so just by sending email and being friendly, I worked my way up the chain and found myself in Hawaii 2 years ago, talking to the director of the Reef Assessment and Monitoring Program. And I said, you kind of got a problem here, because how are you going to monitor all these 14,000 plates? There's a lot of good information in them, and the world needs them. Please, may I have a copy of every photograph and all the environmental data you can possibly get together? I want to build deep learning models to analyze them. And he said yes. He and I are now friends, and I'm really happy that he said yes. The project was in pretty good shape, and then COVID hit and kind of hit the pause button. We call it Coral Vision. And the aim is to do deep learning analysis of these 14,000 plate photos, to measure the diversity of the plates, to estimate diversity at the reefs, and correlate that with environmental data, stuff like acidity, salinity, temperature, to figure out what's driving the processes that cause a reef to crash, and maybe predict at-risk regions. Another piece of this project is citizen science. So I want to create a website where people can do their own analysis, do their own hypothesis testing on these 14,000 plates. And then also, since by then we'll have a deep learning model to figure out what's on a plate, people who've got their own plates will be able to upload and analyze their own plates. So this is one of my projects. It's a source of extreme satisfaction for me, because I get to wake up and think, okay, today I get to work on making the ocean healthy. But corals aren't the only things that build reefs in the ocean. Sponges do it too. They are ecologically important, especially during climate change, because they also build reefs. And they also harbor gazillions of juvenile fish. They also do something else, and I'll show you a picture of it later, because it'll probably blow your mind. So like corals, they're ecosystem engineers. Unlike corals, they process water. That is, they filter out nutrients and inject those nutrients into the ecosystem 10 times higher than anything else we know. And when the conditions are right, they form huge reefs. This map's a little hard to read, but if you look at the inset, that's British Columbia there. So do they really process water that fast? Yeah. You can swim up to a sponge and inject a non-toxic dye at its base. And about 10 seconds later, it looks like this. There's the dye being squirted out really strongly from the top of the sponge. Now this looks to me like a barrel sponge, and from the looks of it, it's probably, well, you can't see my arms. If you make a hoop with your arms, it's probably that big around, and it's probably somewhere between 4 and 7 feet tall. The skeletons of these sponges are biosynthetic glass. There's silicon in the ocean, and the sponges filter these silicon atoms out of the ocean, and they use it to make their skeletons. So if you've ever squeezed an ocean sponge, not a SpongeBob kitchen sponge, but the other kind, it's kind of crinkly when you squeeze it. And the reason it's crinkly is that these glass skeletons are being snapped. These structures are microscopic. To me, it's amazing that anything living and non-conscious can build something this geometrically beautiful. So here's the thing about glass that makes it different from normal bony skeletons. Glass lasts almost forever. When something in the ocean dies, its ordinary skeleton is gone within weeks. When a glass sponge dies, its skeleton sticks around for millions of years. Now, when sea temperatures rise, here's how I like to think of it: protect the corals, but bet on the sponges. It may be, if we only knew more about sponges, that we could figure out strategies for creating synthetic man-made sponge reefs. There are many efforts to create man-made coral reefs, but what if those are going to have trouble surviving the acidity and temperature of the oceans until we can get all that under control? Maybe sponge reefs will be a good backup, but we need to know a lot more about sponges. So up here in the Bering Sea, you can see on the right, that's Alaska. On the left, that's far northern Russia. And one of my colleagues goes there. He's way tougher than me, and he extracts core samples from the bottom of the ocean in this very cold part of the ocean. And these core samples go back 3 million years to the early Pliocene warm period, which is the last time that the climate was changing like it's changing now. And if we could study many of these core samples, you know, the core is just like a cylinder. Think of it, it's like a salami. And every slice of the salami is a different time point. And if we could slice up our cores, just slice that salami and take all our different time points and analyze them, we'd have a lot of information about the right conditions for sponge reefs. The problem is, see that black bar that says 1,000 microns? So if the core is about this big around, how many of these are you going to see in every slice of the salami? A lot. Well, the typical plan is you take your core and you put it on one end of a microscope and you put a graduate student on the other end and you have them write down what they see. You've probably figured out what I thought we should do. I think we can do it better, faster, smoother. There's a piece of technology, pretty expensive. I'm looking for a grant to buy one, called a flow cam. You put some samples suspended in water at the top of the flow cam, and the sample goes through a funnel, tiny micro funnel. And what comes out the bottom are drops of water, and each drop contains one particle. And each drop falls past the camera. And so what you get is high quality, very high speed, very high volume photographs of everything that was in your sample. So we can do deep learning analysis on all those photographs. So the plan is to develop a machine learning model that can recognize and classify sponge particles. Once we can recognize them, distinguish them from the grit and the dirt and the other stuff, then we can count them. And we can see how sponge populations have increased and decreased, over time, 3 million years ago. We can analyze millions of photos of individual particles. Because the nice thing about deep learning is once you've trained your model, using it to analyze any individual sample is quite fast. So we can determine the sponge response to rapid climate change. And so here's my crazy idea. What if man-made sponge reefs could be made to protect marine life until we get climate under control? Lastly, microplastics, and I'm not going to show you large-scale slides because it's heartbreaking. There are currently no good assays to determine the plastic content of a water sample. These are microphotographs, and the plan is to use a flow cell camera to capture and classify individual images, pretty much like we were doing for sponges. Well, that's about it. I do want to give you a takeaway slide. Hope through engineering. That's the takeaway. I can't do it alone, but I can do it with you. Keep up your spirits. Documentaries about the world love making you disappointed and discouraged. Don't let them do it. Don't let them make up your mind. Make wise choices, stay away from single-use plastic. And I want to leave you with the one main thing I've learned, that life on this planet is a balance. And yes, when the balance is destroyed, it destroys. But what I want you to remember is that when the balance is protected, it protects. Thank you very much. I'll stop here and I'm open for questions.
Thank you so much, Professor. It was very enlightening to see how we can leverage data to save the environment, specifically the biodiversity hubs of our ocean. We have a few questions we were hoping to ask you today, and I would like to reintroduce Isita for Q&A.
Hello.
Go ahead, Isita.
So the first question we have for you today from Zuka is, why is ARMS used instead of just taking samples?
If you even touch a coral reef, you can kill hundreds of the tiny little living things that keep it alive. The arms plate surface is a bit rough. And so life, and especially invertebrate life, kind of moves in and commits to living there. So we leave it for a year and a half and it becomes an ecosystem. And that way we don't have to destroy part of an existing ecosystem in order to measure the space.
Okay, that makes sense. Moving on to our next question. What do you think the future implications of such research would be on our ocean life?
Well, if it's just machine learning analyzing photographs, there really aren't very many implications. The thing I hope to do is use what I produce and what my colleagues produce to influence policy. And when governments honor science, they're very open to wise use of policy. There are limited resources, so science can advise, this reef looks okay, but it's endangered. Give it some resources. This reef looks okay, but it's not endangered. You can leave it alone. So I think policy is the next step. That's why I love working with NOAA, because they do both research and policy.
That's exciting to have an actual impact in that way. Another question from the chat from Anna Mendez is, how many pictures are needed to teach the system well enough to work independently?
Really, really good question. Very vaguely, you'd like to have at least a couple thousand training images. And of course, more is better.
Okay, and finally, I guess would be, how can students get involved in such research projects?
Really good question. I'm going to type my email address into the chat. I can take on research students who are either Masters of Computer Science, Masters of Bioinformatics, or undergrads in computer science. And I'd be very happy to work with them, as some of our conference hosts can tell us.
Yes. Okay. I guess we have a more informal question, but thank you very much for your presentation. That about sums it up today, and thank you for your time and the knowledge you've shared with us. I'm sure people are more inspired to have an actual impact using their skills. Now we will have a quick break for lunch, and we will be starting promptly at 1:15 p.m. with our next speaker. Thank you.
Bye, everyone. Thank you, everyone.